Snakes are among the most fascinating and unique creatures in the animal kingdom, known for their elongated, flexible bodies, which can stretch to remarkable lengths. One of the key factors behind their distinct structure is the large number of vertebrae in their spine, ranging from about 150 to over 400 depending on the species. Interestingly, despite their continued growth throughout life, a snake never grows new vertebrae after birth. The number of vertebrae is fixed by the time a snake hatches or is born, but as the snake matures, these vertebrae enlarge along with the rest of its body. This process of growth without the addition of new bones raises important questions about how this fundamental feature of a snake's anatomy is determined. Traditionally, it was assumed that the number of vertebrae and many other characteristics of an organism was entirely predetermined by its genetic blueprint, a perspective known as genetic determinism. But as we will explore, the story is more complex. Recent research in the field of epigenetics has revealed that while genes lay the groundwork, environmental factors also play a significant role in shaping how traits develop. Today, we will delve into the fascinating interaction between genetics and the environment, using the development of vertebrae in snakes as a case study to explore the limits of genetic determinism and the far-reaching potential of epigenetic influence. For much of modern biology's history, genetic determinism has shaped how we understand the development of organisms and the inheritance of traits. According to this viewpoint, genes serve as the ultimate blueprint, dictating every aspect of an organism's development, from its physical form to its behaviors and disease susceptibilities. This suggests that once a genetic code is set, the developmental outcome is essentially inevitable. Traits are thought to be pre-programmed, and external influences are either secondary or irrelevant in shaping an individual's characteristics. The idea of genetic determinism paints a rigid picture of biology where genetic sequences alone control the final outcome. However, recent advancements in epigenetics have exposed the limitations of this viewpoint. Epigenetics demonstrates that genes do not operate as independent rulers. Instead, they are highly responsive to environmental cues and signals. These signals can dramatically modify how genes are expressed, introducing a layer of flexibility that was missing from the genetic determinism model. Even fundamental features of an organism, such as bone structure, which we might assume to be fixed, can be subject to epigenetic influence. Epigenetics reveals a complex interplay between genes and their environment, showing us how an organism's traits are shaped by more than just DNA. Today, we will explore the limits of genetic determinism by examining how epigenetic mechanisms affect the development of vertebrae in snakes. Through this example, we'll see how core features of an organism's anatomy, once considered exclusively determined by genetics, can be influenced by environmental factors. This case will illustrate how far-reaching epigenetics is, not only in shaping individual development, but also in driving evolutionary change. To understand this shift, we first need to clarify what genetic determinism proposes. In its traditional form, genetic determinism suggests that genes are the primary, unchangeable blueprint for any organism. Once set, these genes are responsible for everything, every trait from physical appearance to behavior and even disease susceptibility. In this view, the DNA sequence is final and untouchable. It dictates the organism's fate in a manner that is largely immune to modification. It suggests that the number of vertebrae a snake or any other organism has, for example, is entirely determined by genetics, and no external factors can alter the developmental path once the code is established. But is this the whole story? Epigenetics presents a challenge to the deterministic model by showing that biology is far more dynamic and responsive than we once thought. Instead of following a rigid path, gene expression can be adjusted based on environmental inputs, which means that developmental outcomes are not set in stone. This opens the door for significant modification in traits, based not on mutations in the DNA itself, but on how and when specific genes are expressed. Epigenetics provides the missing link that helps us understand this flexibility. Rather than focusing on genetic mutations which alter the DNA sequence, epigenetic mechanisms modify gene expression without changing the genetic code. These modifications can be temporary, occurring in response to immediate environmental conditions, or they can be heritable, passed on to future generations. This ability for the environment to regulate gene expression introduces a powerful new force in biology that genetic determinism could not account for. How does epigenetic regulation work? There are several mechanisms that control how genes are expressed. One of the most well-known is DNA methylation, where methyl groups are added to certain regions of DNA, often silencing the expression of the gene in that region. Another mechanism is histone modification. Histones are proteins around which DNA winds. Changes in histone structure can either condense the DNA, making genes less available for transcription, or loosen it, making gene expression more likely. Non-coding RNA also plays a role in gene regulation, with RNA molecules silencing or promoting gene activity without encoding proteins themselves. These mechanisms are the biological tools that allow an organism's genome to be flexible, responding to environmental cues in ways that can have profound effects on development, survival, and adaptability. Nowhere is this more evident than in the development of vertebrae in snakes. Snakes are a fascinating example of how epigenetics can challenge genetic determinism. Snakes have elongated bodies with a large number of vertebrae, ranging from around 150 to 400, depending on the species. If genetic determinism were fully accurate, we might expect every snake within a species to have the same number of vertebrae. The traditional genetic view suggests that Hox genes, which control body segmentation during development, would dictate a precise number of somites, the embryonic precursors to vertebrae. Under this model, the number of vertebrae would be fixed based purely on genetics. But what we observe in nature is different. Even within the same species of snake, there can be variability in the number of vertebrae. This points to the fact that something more dynamic is at play. Epigenetic factors, like environmental conditions during a snake's development, can influence the expression of Hox genes and other regulatory pathways, subtly altering how many vertebrae actually form. For example, factors such as temperature, nutrition, or stress levels experienced by a mother snake during pregnancy can influence the developing embryo, affecting the rate at which somites form. Epigenetic modifications, like changes in DNA methylation, can adjust the timing of gene expression, leading to variation in vertebrae numbers even within a genetically similar population. This flexibility highlights the power of epigenetics. The genetic blueprint provides the foundation, but the environment plays an important role in modifying the outcome. This shows that traits like vertebral number, which genetic determinism would assume are entirely pre-programmed, are actually dynamic, shaped by an interaction between genes and the environment. This example challenges the rigid assumptions of genetic determinism, revealing that development is not fully fixed. The snake case study also exposes the limitations of genetic determinism as a framework. It assumes that traits are immutable once determined by the DNA sequence, but epigenetics proves that genes are far more responsive to environmental signals than we previously thought. Gene expression is dynamic, and epigenetic mechanisms allow organisms to adjust their development in response to external conditions. This flexibility introduces a plasticity to development that allows for different outcomes, even with the same genetic code. From an evolutionary perspective, this plasticity provides a major advantage. Epigenetic flexibility allows for rapid adaptation to changing environments. If, for example, vertebral number or structure provides an adaptive advantage in a particular habitat, epigenetic modifications could enable this trait to emerge quickly without needing genetic mutations. This challenges the deterministic view that evolution depends solely on random mutations in the DNA, showing instead that epigenetics can accelerate adaptation by modifying gene expression in response to environmental pressures. This leads to a profound realization. Evolution is not purely driven by genetic changes over long periods of time. Epigenetics offers a more flexible mechanism for adaptation, allowing populations to respond to environmental shifts within a few generations. This ability to adapt through gene regulation introduces a faster, more efficient way for traits to evolve, transforming our understanding of both development and evolutionary biology. The study of epigenetics, especially in cases like the development of snake vertebrae, exposes the limits of genetic determinism. It reveals that while genes provide the instructions, how and when those instructions are carried out depends on environmental signals and epigenetic modifications. This more nuanced understanding of biology underscores that organisms are not solely the products of their genes, but of a continuous and dynamic interaction between their genes and the environment. Epigenetics shifts the paradigm, showing that traits long thought to be rigidly fixed, such as skeletal structure, can be malleable, shaped by both inherited and external influences. This flexible view of development and evolution opens new avenues for research and deepens our understanding of the complexity of life. This raises an important critique of genetic determinism. If something as fundamental as bone structure can be influenced by epigenetics, what other traits might be subject to environmental regulation? Could traits like human intelligence and consciousness be shaped by epigenetic factors as well? Emerging research suggests that this is not only possible, but likely. Intelligence, for instance, isn't strictly the product of our genetic inheritance. Studies show that environmental factors, such as early childhood experiences, prenatal nutrition, and stress, can alter the way genes related to brain development and function are expressed. These changes, brought about by epigenetic mechanisms like DNA methylation or histone modification, can affect how neurons connect and how the brain processes information. The idea that intelligence or consciousness could be regulated by epigenetics opens a door to an even deeper question. Could these traits be switched off under certain conditions? If epigenetic changes are reversible, then could environments that are deficient in necessary nutrients or high in stress diminish cognitive abilities, just as they might alter bone structure in a growing snake? And if so, what are the broader implications for humanity? This line of thought raises significant concerns about the environments we create for ourselves, particularly in extreme settings like outer space. As we look toward the future and the possibility of off-planet survival, the stakes become higher. The environments in space, marked by radiation exposure, microgravity, and isolation, could influence gene expression in ways we are only beginning to understand. Radiation, for example, could induce epigenetic changes that lead to long-term health issues like cancer or even cognitive impairments. Microgravity affects how bones and muscles develop and may influence gene regulation related to skeletal health. Psychological stresses, such as isolation and confinement, might lead to epigenetic changes that affect mental health and cognitive function. These concerns point to the need for a more deliberate and thoughtful approach to environmental design in off-planet habitats. If our environments can shape our biology at such a fundamental level, then ensuring that future space habitats protect and promote healthy gene expression becomes a critical task. It's not just about shielding astronauts from the physical dangers of space. It's about creating environments that support mental and physical well-being through careful attention to the epigenetic factors that influence development. And even here on Earth, understanding the profound impact of epigenetics invites us to rethink how we interact with our environment. If our surroundings, whether that's the food we eat, the air we breathe, or the social conditions we live in, can shape something as core to our humanity as intelligence or consciousness, then the responsibility to protect and nurture those environments is paramount. Public health initiatives, social policies, and even educational systems could benefit from incorporating epigenetic insights to ensure that future generations are born into environments that support optimal development. Ultimately, what the study of epigenetics reveals is that traits we once thought were immutable are, in fact, subject to change based on the world around us. Genes are not destiny, they are potential, shaped by a complex interaction between biology and environment. And as we push the boundaries of human exploration, whether that's into space or simply into new realms of scientific understanding, we must take seriously the role of environment in shaping who we are and who we might become.